All right, good evening, everyone. Welcome to Tuleyome's January Nature and You lecture. My name is Jeff Ben. I'm the Education Associate at Tuleyome. If you don't know, Tuleyome is a conservation nonprofit based in Woodland, California. Our mission is to engage in advocacy and active stewardship with diverse communities to conserve, enhance, restore, and enjoy the lands in the region. The region we focus on is the northern Inner Coast Range and neighboring areas of the Central Valley, and we especially focus on the Berryessa Snow Mountain National Monument, which we helped lead the effort to get established and then to get expanded just last year. In addition to that public lands advocacy work, we also do a variety of education programs, including K-12 field trips, natural history articles in our newsletter and local newspapers, and of course, this monthly lecture series. We also run a range of adventures programming, including hikes, trail building days, wildflower tours, and our youth boot bank, which provides free loans of hiking boots to kids. I also want to give a special plug for our Spring Thing event, which is coming up on March 22nd at Woodland Regional Park Preserve. This is our big annual event, which features nature-related tours and activities for kids and adults, live music, a food truck, and lots more. So be sure to come check that out. If you'd like to learn more about these or any of our other projects or upcoming events, you can do so at our website, tuleyome.org. On to tonight's lecture. This lecture series, like all of our programs, are supported in part by donations from folks like you. So if you're able to and haven't done so already, we gratefully appreciate a donation to help support all of the good work that we do. So tonight we are very lucky to have Dr. Julin Maloof and Dr. Jenny Gremer as our speakers. Dr. Maloof earned his PhD in biochemistry from UCSF and is currently a professor of plant biology at UC Davis. Dr. Gremer earned her PhD in organismal biology and ecology from the University of Montana and is currently an associate professor of evolution and ecology at UC Davis. Tonight, they're going to be talking to us about their research on California jewelflowers. During the talk, we ask that you keep yourselves on mute and hang on to any questions you might have until the end, at which point we'll have time for some Q&A. All right, Dr. Maloof and Dr. Gremer, take it away.
Thank you everyone for coming. We're really excited to be talking to you about what I think are a really interesting group of native California wildflowers called the jewel flowers. And so we'll be talking about, it's all about timing, the causes and consequences of germination timing in California jewel flowers. Today on our work in those beautiful California jewel flowers, which includes Streptanthus and Colanthus species. We refer to this as the Streptanthoid complex, but it's a group of what I think are really fascinating native wildflowers that are in the mustard or Brassicaceae family. On the slide here, you can see some of the species and their beauty and diversity of form and flowers. And hopefully, maybe some of you have seen and maybe recognized some of these beautiful jewel flowers that we have here in California. Next slide. This group is part of the ancestral Madro-Tertiary flora, which originated in the southern deserts. You can see that illustrated in the graph here, where we've got those southern deserts in southern Arizona and Mexico and Baja. And then plants migrated northward as the climate warmed and dried. And so this flora essentially evolved in conjunction with California's dry Mediterranean climate. Next slide. So the Streptanthus clade spans the California floristic province, which maybe many of you maybe already know this, is a world biodiversity hotspot because of that high diversity and a high level of endemism, which means that there's a bunch of plant species that are only found here in California. And so California is a great place to study and enjoy plants and to do botany because of this high floristic diversity. On the left here is a phylogenetic tree for the Streptanthus group, which includes Streptanthus and Colanthus. I'll talk more about that in a minute. But what I want you to see here is on the graph showing the map of California. And each of these colors is a different species. The C stands for Calanthus, the S stands for Streptanthus. And so the different points are colored by the different species, and then kind of the shapes are drawn around where those species have been found in California. So what you can even see just from this overlap of colors and the coverage in California, we see that this group of jewel flowers really covers a wide range of the geography of California, which we know also means a whole range of different climates and habitats. And we have a bunch of diversity in those habitats and climate as well. So this slide zooms in on the phylogenetic tree, the phylogeny. And so this was developed by work by Sharon Strauss and Eva Lou Caucho, who have been working in this group of species for many years. And so this is a phylogenetic tree, which illustrates the evolutionary relationships among the species. So it's a lot like a family tree. So the tips on each of these branches is a species, either Streptanthus or Calanthus with those same abbreviations, with the species epithet spelled out at the after that. So what you can see from this is like the pattern of branching reflects how the species evolved from a common ancestor, with each branch indicating which species diverged from a common ancestor. And species are more closely related when they share a more recent common ancestor. So kind of like we think about with a family tree, that's what those branches tell us. So what Sharon and Evalu did was use this to systematically group species by those relationships, and those groups are shaded in with these different colors. So through Evalu and Sharon's work, we learned that this whole group, like much of the Madro-Tertiary flora, has desert origins and diversified into northern climates. Um, let's see. These species are so essentially those Calanthus species, we're missing a label, but those at the bottom are species from that Mojave Desert, Carrizo Plain, Central California. That next Hoalii alliance, that group in the are kind of pink there, or more of our high elevation perennial clade. And then we move up into the blue where we get those Northern California and our Coast Range species. So these are probably the Streptanthuses and or Calanthuses. I think it's all Streptanthus in there that you may see on Tuleyome lands or associated areas. And then in the kind of more orangey on top is our transverse range to Northern Sierras. So they really have this like started in the desert and then evolved into these more northern, more kind of mesic Mediterranean climates. At least they're mesic when you compare them to the desert, right? So, most of the species in this whole group are annual plants. And so that means that they complete their entire life cycle within one growing season. So, from germination to seed production is all within one season. There is that high elevation perennial clade, that Howellii alliance in that kind of pink, more pinky color, that has Longicolicus and Shastensis and Howellii. Perennial means that they live for more than one growing season. So with plants, this is a big difference between living for one growing season or multiple growing seasons. And then what I think is even more interesting is that some species, including one that Julin and I study very intensively, has facultative perenniality, meaning that they sometimes express an annual life history and get it all done in one growing season, and sometimes they live for multiple years, which we're trying to still figure out how that works. Um, okay, next slide, please. Now, these are just gratuitous photos of these adorable jewel flowers that you may have seen while hiking on Tuleyome lands or in the coastal ranges. Actually, Jeff, I think you said you saw glandulosis recently on a hike. And then we also have Streptanthus morrisonii, Streptanthus breweri, and Streptanthus drepanoides. Have folks seen any of these species while hiking around Tuleyome lands or in the Coast Range or various places? Maybe some of you. Great, awesome.
See a few hands going up, yeah.
Yeah. um, great. And then the next slide, please. So these beautiful wildflowers, I would call both delicate. They have these amazingly delicate flowers, but they're also really hardcore. So they're a really interesting group to study because they've adapted to different aspects of California climate as they diversified across the state. They're pretty stress tolerant. They live in bare, dry, and sometimes fairly inhospitable soils. Um, so they're hardcore in that way, but they're also terrible competitors. So they get excluded from other areas because of competition from other species. So they're kind of also good at living in those dry areas, and that's kind of where they are able to stay there. Um, Sharon Strauss and Evalu also studied how they have had this adaptation to bare environments, and that kind of preceded some of these really interesting habitats we see them in, which includes these really mineral-y soils and serpentine soils. Maybe some of you have heard of serpentine soils or have seen them as you've been hiking around the area. So that's soil that's characterized by really low nutrient content, low water holding capacity, and often having heavy metals, which can be toxic to many plants. But we have many of our streps can live on serpentine soils, and there's several of them that have actually adapted to live only on those serpentine soils. So again, I think that's fairly hardcore. They also have really interesting cryptic coloration, like you can see on this slide here, and maybe you've seen them out in the field. They can be hard to see because they can be kind of these dark purple to mauve to brown colors where they like basically are really hard to see against the rocky outcrops in the bare soils that they live in. Um, we also know that there's several threatened and endangered species in the group. So again, a little bit delicate in some ways, right? And my sense is mostly those are threatened by kind of habitat destruction and fragmentation. But we also expect to see some effects of climate change on the group as well. Okay, next slide, please. And here I'm just showing some examples of Streptanthus habitat, which includes like rocky and sandy habitats with a variety of parent material, so a variety of different types of soil and or amount of soil, right? So we see them on limestone, we see them on shale, gabbro basalt, serpentine, sandstone. We often see them on these slopes like this. So sometimes I joke that Streptanthus, like finding Streptanthus is sometimes like rocky outcrop or like roadside ecology. Um, and so, yeah, so they tend to kind of live on the edge of those environments that other species like to live in. Okay, so for these jewel flowers and many other plants in changing environments, really timing is everything, right? So getting life history timing right can mean the difference between success or complete failure. And so we think a lot about germination timing. That's what we're going to be talking about with you today, which is this critical transition in which a plant goes from the relatively protected seed stage to the incredibly vulnerable seedling stage. So it's crucial to time that transition with favorable conditions for establishment. And this is particularly true, I think, for annual plants that live for only one growing season. So seeds are the only link to their future. And once they germinate, they only have one chance, right? So that makes that transition really important, and getting that timing of when to germinate right, even more important. We also know that germination timing has really important consequences for that individual's performance, and that it kind of sets up the life cycle for the plant. So it kind of sets up what the environment that establishing seedling is going to experience and what the like older mature plant is going to experience later in the growing season. Um, and so, oh, and click please, Julin.
Right.
Similarly, reproduction is really important, right? So the timing of reproduction and the timing of flowering is really important. And for our jewel flowers, which are primarily outcrossing, which means they primarily need pollen from another plant in order to produce seeds, they need to get this timing right so that there's pollen available from other plants, right? So, and that there's pollinators around to bring that pollen to them and to move their pollen to other plants. So getting that timing right is also really important. So we know in California, we have a really variable environment. Every winter, water year seems to be really different when it starts, how much water we get, how warm or cold it's going to be. And so really getting that timing right is a real challenge. And so we've been really thinking about how these jewel flowers do that. Okay, and the next slide. And we know that climate change is really altering conditions and the cues and conditions that plants experience. So they're seeing shifting seasonal conditions. So on the graph here on top, I'm showing just global annual temperatures throughout the year. The different colors are different years, right? And as we know, right, temperature is getting warmer every year. Um, this graph on the bottom shows us a little bit more about the changes in precipitation that we're experiencing in California. So these are changes we are already seeing since pre-industrial times. So what the different lines here indicate are just different latitudes in California. So we span a lot of latitude in this state. And what you want to look at is the values below zero means that there's been a decrease in precipitation relative to pre-industrial times. Patterns above zero means there's been an increase. And so what we see from this work by Daniel Swain et al. Is that we're really seeing a shift, especially in fall precipitation, where we're getting less precipitation in the fall. And when that precipitation is coming later. Other papers have also shown that it's coming about a month later than it used to. And so that means we're having a shift in precipitation timing. There's less in the fall. It's becoming more kind of concentrated in those winter months, and then we also have less in the spring as well. So really our growing season conditions are changing, and that means that timing is changing. And so these jewel flowers are dealing with a really shifting environment and needing to get that timing right as it changes. Over to you, Julin.
Okay, so as Jenny has set up so nicely, timing is everything, and the correct time to germinate or reproduce may be changing as the climate patterns change. And so I'd like to spend a little time discussing what we know about how does a seed time its germination? How does it get it correct? And I'm going to ask for a little audience participation here, so you can, if you want to respond to my questions, you can just type in the chat. But just kind of from what you know about plants and plants getting established, what would you say is important for seedling getting established? So the seed germinates, and then what does it need to have the seedling get established and thrive? So if you want to type in the chat, feel free to go ahead. Got moisture and soil temperature? Anybody want to add anything to that? Correct amount of water, yeah. Okay, so yeah, so those are great. I would add to that light and kind of nutrient conditions, and there's probably more. So the first part of timing is simply, I don't know if simply is the right word, but I'll say simply, are the conditions at the time good? Is there sufficient water? Is the soil temperature good? Is there light? Are there nutrients? And so seeds have the ability to sense all of these aspects of their environment. And so that's one part of timing, is just our conditions, are the right parts of the environment present to promote seedling establishment. So that's one part of getting timing right. But perhaps more interesting is the annual timing, the seasonal timing, which really Jenny was getting at. And so I'm going to ask again, if you think about a not just establishment, but for an annual plant, it needs to make it all the way through to reproduction. And so actually, maybe I'll just keep going on that. So what does an annual plant need to be successful? So it needs to germinate at a time that it can complete its life cycle with sufficient water, with nice growing conditions, etc. And so this is, to kind of bring back up what Jenny was saying, timing is really critically important. And so I'd like you to think about a plant that grows in the alpine environment, an annual plant like Streptanthus. Some of our species do grow in the alpine environments. And so for these species, they produce seed in midsummer. And so the seeds produced, they don't have good conditions to germinate then because the soil is usually pretty dry. But then it'll start raining, and it'll rain in October. And so now, like, all of those kind of immediate conditions maybe are pretty good. The soil might still be warm, there's water, there's light, there might be good nutrients, but that is a bad time to germinate, right? Because what'll happen? If you germinate in October and then it snows, and you have a little tiny seedling that's buried under 5 or 10 feet of snow and has to survive under that snow for 6 months. That's not a recipe for success. What strategies do plants or seeds use to avoid that fall germination? And one thing that they do is they basically some species and some populations require extended exposure to cold before they're actually will germinate. And this process is known as stratification. And so from what I've told you, you might hypothesize, and Jenny and her colleagues hypothesized that the stratification requirement might be different for alpine populations. And so this is an interesting question for our research groups in general, because as Jenny said, like, we're really interested in how do plants adapt to different places in California. So Jenny and her colleagues hypothesized that the stratification requirement might be higher for alpine populations. Those populations, they've got a long cold season, they want to make sure that they don't germinate too early. Whereas populations or species that are native to lower elevations, they're exposed to less cold. So if they have too strong of a cold requirement, they may not germinate at all. And also for low elevation populations, it can actually be advantageous to germinate in the fall. So maybe they shouldn't have any cold requirement at all. So that was the hypothesis. Jenny and her colleagues set out to test this, and they tested it in Streptanthus tortuosus, one of the species we studied. And the experiment was like this. They got seeds from populations that were native to different elevations, and they then gave those seeds different amounts of chilling or stratification, and then they measured the proportion of seeds that germinated. And so starting just from the low elevation populations, what we can see is that they germinate. It doesn't really matter what you do. Give them no chilling, give them a lot of chilling, they're going to germinate. And so this is consistent with those low elevation populations. A good time for them to germinate is in the fall. It rains, they germinate, they grow over the winter, everything's good. But if we look at the high elevation populations, what we see is that in order to get them to germinate, you see that most of the ones that are germinating have black dots. So they've had the 7 weeks of stratification. So for high elevation populations, they require extended chilling to germinate. And so this shows then one way in which the seeds are like interpreting cues in their environment and how that has allowed them to adapt to different environmental situations. Okay. I'm not going to go into as much detail, but I'll just say that there is also a similar process that can control flowering season. Again, it can be disadvantageous for alpine plants to flower in their first growing season because there's not enough time to complete their full life cycle. And so similar process, some plants require a long period of exposure to cold before they will flower. And so that process is known as vernalization. Okay, so that gives you a little bit of an idea of how plants are adapting to different climates across the state. And that kind of sets up the the research questions that our groups are interested in. So we're interested in that specific question: how do plants adapt to different environments? And I've shown you a little bit of that. What are the different life history strategies they use? That is, how do they time their life cycle transitions? We are also interested in understanding physiological differences. So how do plants change their growth to be able to deal with hot, dry environments versus cooler or wetter environments? And also, what are the genetic mechanisms that cause these changes, right? So these plants are different. What are the genes that change to allow those differences to occur? We are very interested in studying this in the context of current and future climate change. So timing's important. These adaptations are important, but everything's changing. So how does that impact plants that have specific adaptations to certain climates? And if we find that there is an impact, can we develop some strategies to help mitigate the impacts of those climate changes? And I think now, Jenny, it's back to you.
Yes. Thanks, Julin. And so we've been I had a conversation with someone that said, basically we've been interviewing seeds and interviewing these species to understand how they're sensing their environment, how they're responding to these changes. And so we've done a series of studies to understand this. And so in a first study, we asked, how does that shifting rainfall timing affect germination? And so remember when I was talking about California's changing climate, right? Our precipitation, our first rain events that'll trigger seed germination are coming more like end of October if we're lucky, right? Sometimes not even until November and December some years. And so on average, across the last 30 years or so, our germination triggering rain events are happening later. That's all set up against a backdrop of, right? They vary a lot from year to year anyway, right? So these plants experience very different germination timing in different years. And so we wanted to ask, how does this influence these plants? So what we did in this experiment is we set up a study in a screen house, which is like a greenhouse with a solid roof, but the walls are screens. So that exposes plants to those ambient seasonal conditions, those seasonal temperatures. So in this way, we could simulate different timing of germination triggering rain events while keeping the realism of the temperatures that seeds would experience. And so I'm showing that on the graphs here. So on the left, on the x-axis is date, on the y-axis is mean daily temperature. These were the conditions in our screen house. The different dashed lines indicate the rainfall events that we simulated. And so we started those from September through January. And then the graph on the right also just shows what those temperatures are. And I kind of like showing both of these, right? So we can see that those earlier germination events at the top of the graph in 17th of October or of September was much warmer than our rain events that we simulated into December, right? So later rain means colder temperatures that these species are germinating under. Julin, I think there's an animation there.
Oh, yeah.
Just go through all of them. And so what I have highlighted here in the red and the blue, the red is the historical mean onset date that climate scientists have identified from 1960 to 1989, those rain events came earlier. Nowadays, they're coming more later in October, at the end of October. That's those blue boxes in both of those graphs. So later rain means cooler temperatures these species are experiencing. Okay, next slide. So we wanted to ask how these different species are responding to that shifting rainfall time, both the variability in rainfall timing and whether there was differences with rainfall coming later. So there's a bunch of graphs here. And on the x-axis, I have what I call the cohort planting date. That's the time that we simulated those germination triggering rain events for each of those experimental groups. And then on the y-axis is germination proportion. So 1 means 100%, 0 means none of them. And so what you'll see here, this is for Streptanthus drepanoides, these are 4-letter species abbreviations, is that our germination proportions were lower as the rain events came later. And so we found that for many species. Next slide, please. And so what we see is that 8 of these 11 species that we had in this experiment, which includes a bunch of Streptanthus and Calanthus species across the phylogeny, 8 of them germinate significantly less when rainfall comes later. Next slide, please. Those 3 that didn't were our species from the drier, more variable environments, and they're not that sensitive to that changing rainfall timing. So these are our desert species, our really dry adapted species that have low germination rates because they're spreading germination over multiple years because they're worried about germinating in the wrong year. And so they were a lot less sensitive to this rainfall timing, and that might be because they've adapted to a really dry, variable desert climate. Next slide, please. So here we're just highlighting, so those red dots here indicate that historical onset, which is that October 2nd, versus what we're seeing with climate change, that onset at October 30th. For these 4 species, we saw significantly different amounts of germination across those 2 dates. So that suggests that those 4 species are probably already significantly impacted in terms of their germination proportions to the climate change we've been experiencing in California. I'll note that this tends to be our more northern, kind of wetter, cooler. It's all relative because it's still in California. It's still dry and warm in a lot of these places, right? But these are our less deserty, more northern, wetter, cooler species. Next slide, please. Okay, so that's how they responded in terms of germination with that different timing of when that rainfall is coming throughout our winter wet season, right? But what does that mean for the rest of the growing season and life cycle? So to do this, we did the next experiment, which is we then force those seeds to germinate at different times, because you know from the previous studies that sometimes if we try to germinate them in December or January, they would not germinate. So we forced them to germinate in growth chambers, and then we planted them and we put them in that same screenhouse where they're experiencing those ambient seasonal temperatures. These lines here also illustrate when we forced those germination events, so when each of those groups started their growing season for the year. And it also shows the mean daily temperature and how it changed throughout the season, right? So we had kind of somewhat warm fall conditions that got cooler as we moved into winter and then warmed back up as we moved into the spring and early summer. So we did this for 10 different species, and we studied what that meant for when they flowered and their reproductive performance. So this graph here shows some of our results that I think are pretty clear and consistent. Hopefully it will make sense once I describe the graph. So on the x-axis here, we've got those different timings, like basically when their season started for each of those experimental groups. On the y-axis, we've got their date of first bud. So this is when the plant started to transition to flowering. Each of these panels is a different species. Those are their 4-letter abbreviations, so Streptanthus drepanoides, Streptanthus breweri, and so on. But what we see across all of these species that live in pretty different climates and habitats in California, they all flower later. Their flowering happens later when their growing season starts later. So when they germinate later, they flower later. But maybe that's just a timing thing. Maybe they sort it all out with that phenology. What does that mean for, making more seeds and having that link to the next generation and having fitness? And that's what we have on the next slide. So here I'm showing those same panels for the same species. We also have that those different germination timings, so our cohorts on the x-axis. And then we've got the number of seeds that they produce for each of those on the y-axis. So what we see here is that there are 6 species of the 10 that produce a lot fewer seeds when they germinate in later cohorts. So that means when germination shifts to later cohorts, they make fewer seeds. Or later timing, they make fewer seeds. Okay.
Sorry, were you
Where do you
ready
go?
for the next slide? Yeah, okay.
Yeah.
Okay, so where are we now? So we've told you that Strepsianthus grows in a lot of different environments in California, that they have specific adaptations to allow them to grow in those environments. That timing is very important, and that the climate change that's already occurring can impact the fitness of these species. I'm really interested in understanding what is the what are the genes, what is the genetic basis for some of the differences we see in these timing events in these species. And so I want to spend a little time on that. What are the genetic pathways that have evolved or changed to allow shifts in germination strategy? And, oh, right, I talked ahead of my slides here. So what we've shown that these species vary in the germination cue response. We know that those changes are heritable from generation to generation. So that means there must be a genetic basis for these differences. And if we can determine that genetic basis, then we understand a little more mechanistically how plants are evolving these adaptations. And so the strategy that we've taken is to correlate germination behavior with something called gene expression, which I'll explain over the next few slides. But it's a way to relate the observed germination behavior to the underlying genetics. So I'm going to give a little background, a little genetic background here about proteins, genes, and genomes. And let me see if I can get the pointer to work. Can you see a pointer now? Okay, great. So let's start talking about proteins. So proteins are basically the molecules that perform most of the functions in an organism. And so just to give a couple of examples that are maybe relatable, proteins can be enzymes that carry out chemical reactions. And one you may be familiar with is the protein lactase, and that is the enzyme that's responsible for digesting lactose. So if you're lactose intolerant, it's because you don't have that protein being present in your stomach or in your intestine as an adult. Proteins can also be structural, like keratin. So keratin is the main component of hair, and it's a major component of skin. Okay, so proteins, structural, and they do things in the organism. Proteins are coded for, the instructions to make the proteins are basically genes, and there's roughly 1 gene per protein. These genes are encoded for in the DNA of the organism, and that DNA is organized into things called chromosomes, which is what's illustrated in red, and all of that DNA together, all the chromosomes, makes up the genome of the organism. Okay. So, what's an important point here is that the genes, the things that code for the proteins, can be turned on or off. So, for example, the K genes that code for keratin, those don't make keratin everywhere, right? They make keratin in the skin cells and the hair follicles. You don't want keratin being made in your stomach. So the keratin genes are on in the places it should be made, and they're off elsewhere. Same thing, lactase, that enzyme that digests lactose, is on in the small intestine. It's off elsewhere. There's also timing here. So lactase is on in infants, but it's off in most adults. Why is this relevant for this talk? Because the gene expression can reflect adaptations and kind of the physiological state of the organism. So I mentioned that lactase is off in adults, most humans. For some humans, lactase is on. Maybe I'll turn it back to the chat for a minute. So but my question for you, if you feel like answering, is which human populations tend to have lactase expressed in adults, and why might this have arisen? Europeans, says Jeff, with less other food in the winter. Yeah, so it basically correlates with populations that historically used cows. Yeah, cows available, exactly. Right, so populations that had cows available for dairy production. And so so cows are a kind of amazing thing, right, for humans. So cows take grass, which we can't digest, and turn it into milk, which at least if you have your lactase gene expressed, we can digest. And so that's a pretty beautiful thing. And so indeed, for populations that were able to herd cows, or yes, goats, sheep, reindeer, that's a very strong advantage. Okay, it actually turns out there's some African populations also that have lactase expressed in adults, in particular the Tutsis and the Beja, who are herded. They have a pastoral history. So the point here is that that's an adaptation, right? So that's the lactase gene being on in human populations that were pastoral. That was an adaptation to that lifestyle and allowed them to take advantage of having cows or reindeers or whatever milk producing animals around. And so then we can take that same, and it also gene expression then can inform us about what genetic pathways are being used to adapt to different situations. So we can take this idea and then apply it to our plant questions. Okay, so again, the actual question here is what genetic pathways have evolved to allow shifts in germination strategy. And so what we did was to measure the gene expression, that is what genes are on or off, or how on are they, for all of the genes that are in the dry seed of the Streptanthus in the studies that Jenny was just talking about. And then that's a lot of data. And so we grouped those genes into sets of genes that behave similarly. And then we asked which sets of genes were correlated with germination, or were there any? So the answer to that is yes. So on these plots, what we're looking at is this gene expression. And we've got two different examples. So the left example, the map is showing you how high this particular set of genes was expressed in different species that were native to different parts of California, with blue meaning higher expression and orange meaning less. And the graph is relating germination proportion on the x-axis to how turned on these sets of genes were. And so this set on the left, it's a set of genes that is high in seeds that germinate readily and low in the seeds that don't. And then there's another example on the right, and this is a set of genes that is expressed strongly in species who have seeds that don't germinate readily, at least in our conditions, and expressed low in the seeds that do. So these genes then may be related to the different germination behavior of these species. We can ask what are the functions of these genes, excuse me. And for those genes that were correlated with ready germination, we found that they functioned in things like promoting growth of the seedling in response to light, in producing energy, in dividing cell division. And in contrast, the genes that were associated with low germination were known to function in processes like inhibition of development, response to heat, or this plant hormone called ABA, which basically is a hormone that when the plant is stressed, it like puts the brakes on. It says, all right, take it easy, don't develop too quickly. And so remember that all of this came from dry seed. So what we're really looking at is the kind of what's the program that these seeds are set up with. So they're in the soil, maybe waiting for rain. And so we get the picture that the seeds that are readily going to germinate when they get rain and have good conditions have a program already that's like, all right, let's go, let's get on it. And the other seeds that are going to be more cautious or less likely to germinate, they're set up with a program that's like, yeah, take it easy. So that for me is pretty exciting finding that we're seeing kind of genetically what's controlling this different behavior in the seeds. Now, these differences could be random. It could be that there's just some, and a lot of evolution is this way. A lot of things that we see that are different in different species, changes just happen randomly. Or changes can be selected for as an adaptation. And so one can test for are the differences random, or are they more likely to be due to an actual adaptation? And so we did that for our gene modules. And without going into details, the answer is yes, that those modules that are related to germination behavior actually show evidence for selection having selected for the different levels of expression. Okay. So now we've told you that not only are there a lot of different adaptations, we're starting to get at some of the genetics underlying that. I want to take another minute or two and talk about, in terms of genetics, how do plants innovate? What is the material by which they develop new adaptations? And so the genes that are already in the genome, those can change and definitely will contribute to differences over evolutionary time, but also realize that the genes that are already present in the genome and have been there for a long time, they have a function and they're constrained because if if there's a mutation that causes them to do something else, they may lose what they're doing that's already important. So basically, genes that have been in the genome for a long time are pretty constrained and slow to change. And so a biologist, Susumu Ono, in 1970 published a kind of landmark paper where he really highlighted the idea that duplicating genes can allow for rapid innovation in terms of evolution. And the idea is that if you duplicate a gene and you've got one copy that can keep doing whatever important thing it was already doing, and then another copy that kind of can be tinkered with and develop a new function. And so we were wondering if there's evidence for significant gene duplication in Streptanthus. And so to address that question, what we did was to actually sequence the genome of one of the species, Streptanthus diversifolius. And what we found was that the whole genome has been duplicated in the entire Streptanthoid clade. And this is illustrated here. So on this plot, each horizontal bar represents one of the chromosomes. And so all of the bars together represent a genome. And I'm showing you two different genomes. So the middle set of bars is from a plant called Arabidopsis thaliana. It's also in the mustard clade. And it has 5 chromosomes. So that's shown here. And then I'm also showing you the diversifolius chromosomes both above and below Arabidopsis, and it's a different set above and below. And then these vertical lines show regions of similarity. So what you can see is that for each stretch of the Arabidopsis genome, there's actually 2 diversifolious chromosomes that match. And so that is basically the illustration of the duplication, whole genome duplication. So while we don't know that this is true, this result gives us the hypothesis that maybe it's this whole genome duplication that has provided the genetic material that allowed the rapid adaptation and diversification of this Trithanthus clade into these different environments, into the different soil types, etc. Okay, and so we're nearing the end, but I'll turn it over to Jenny and I'll come back to me briefly and then we'll wrap it up.
Okay. And Julin, there's some animations here, I think. So, right. So what have we told you so far, right? So with climate change, we're seeing a later onset of those fall and winter precipitation events, which delays germination for many species. We are seeing warming temperatures globally, but actually it turns out that that shift in precipitation timing means that germination is happening later so that it's actually happening under cooler conditions. So like even the like couple degrees of warming that we've had is not enough to make up for that difference in germination timing. But we do know we do want to think about how those warming temperatures also influence other parts of the life cycle, which we won't talk about much here, which is like when and how they flower, right? So we know that climate change is affecting the timing of the life cycle. We saw that later germination triggering rain events, which happen under cooler temperatures, reduces germination for many of our species, except for our kind of deserty, kind of variable, arid, adapted species. We also saw that it's changing the timing of flowering. So when they germinate later, they flower later in the year. And while maybe some of that shifting can compensate for that for effects on fitness, we also saw that those later germination events, those also had lower flowering and they also produce fewer seeds. So it's reducing germination, so fewer seeds are germinating, and they're also producing fewer seeds when they do germinate. So that can be pretty tough on these species in persisting across generations, right? Those seeds are the link. But we did see that there's this real different sensitivity across this group of delicate and hardcore jewelflower species, where we're actually seeing that our more northern, more mesic, Mediterranean species, they're more sensitive to that germination, whether they germinate or not with the later rains, and how much of an impact on fitness and seed production that's producing. And what else have we told you? So Julin told you about how we've seen how there's differences in how they're really primed to respond to that precipitation when it happens, right? That also explains the differences between what we see in those northern species versus our southern desert dry adapted species. And that those modules that are under and have been under selection and suggest that they have evolved as these species have moved into these different habitats. And that a whole genome duplication may have facilitated some of this climate and edaphic and habitat adaptation that these jewel flowers have done as they've diversified across the California floristic province. Next slide, please. So basically what we've been thinking about, right? So we've been seeing how they're responding to already experienced climate change. What does this mean for the future?
So, what does this mean for the future? To try and answer this question, we are focusing on one species, Trithanthus tortuosus, which we've already told you some data about. We've chosen tortuosus because it in one species kind of encapsulates a lot of what is really exciting about this whole group. It makes it good for these kind of studies. It's got a very wide distribution across California. It grows from low elevations to like way up in the alpine. So it's adapted, this one species has different populations adapted to really different climates. And so we see a lot of both within and between population variation in germination timing, in timing of frequency reproduction, lifespan and strategy from annuals to biennials and perennials. So it's a really interesting species and provides a lot of material for trying to understand what's happening in terms of climate change.
Also
And
differences in flower color.
right, and the difference in flower color, exactly, yeah. I mean, in fact, actually, when I was first started working with Jenny and with Annie Schmidt, and I went out to the greenhouse, they had a lot of different tortuosus populations growing. And I was actually stunned to learn it was all the same species. I mean, they just looked, those different populations looked like different species. It's quite remarkable. But it is the same species. We know we can make genetic crosses between them, so they clearly are. So we started studying tortuosus and trying to understand the adaptation of the different populations. And one thing we've been doing is just asking how well are they adapted to their current growing location. And so one study we've done has been focused at Wrights Lake, which is at the edge of the Desolation Wilderness up Highway 50. And tortuosus does grow there. And so one of the experiments we've done there is to just plant different tortuosus populations that are native to different locations at Wrights Lake and ask who survives, like who does best in that location. And so the plot on the left is showing some of these results. And so we've got proportion surviving on the y-axis over time of the experiment on the x-axis. And then each line represents a different population. The arrow points to the native, I mean, we planted these, but it points to seeds from that Wrights Lake location. And it does okay, but it's not the best. So what's surprising here is that actually the 4 best populations all come from lower elevations than Wrights Lake 2 itself. So this points to an adaptational lag potentially. So the idea is that climate's getting warmer, and so Wrights Lake 2, the seeds that are native to the location no longer match the environment there, that actually seeds from lower elevations might match that environment better. And surprisingly, at that location, these plants overwinter before they flower. And surprisingly, some of the low elevation populations actually even did better over the winter, which honestly, I don't understand that, but there it is. Okay, so to finish this up, I want to talk about some strategies for mitigating the impacts of climate change. And people have been constantly working on this forever and have developed a lot of strategies. And so one of the simplest ones you might think about is what we would call assisted migration. So we move plants from a low to a high elevation. So as I just said, it looks like at Wrights Lake 2, at least based on 1 year's, well, really 2 years now of data, that low elevation populations might survive better there. So maybe we could just move those up. That's not really a great strategy because then we lose kind of the endemic genetic diversity that's present in that Wrights Lake 2 population. And, while survivorship, kind of raw survivorship might be better for low elevation populations if we move them up, we're losing things that might also be important for Wrights Lake 2. That could be defense against specific insects that are in that location, or it could be other UV protection because there's more UV there. Anyway, we might lose a lot of things that are important about living at Wrights Lake 2. So another approach would be to do something a little more fine-grained. So don't move whole populations, but do some genetic crosses so that you bring in some advantageous genes from a lower elevation. And so this can be a good strategy. The challenge is, how do you choose the right source population, and how do you choose the right genes? And so this is what we're working on. We have a collaboration to develop basically models that integrate genetic information and ecological models, with the idea that this can hopefully help us predict the best source populations, the best crosses, and kind of the best combination of genes to assist an endemic population that might be suffering from changing climates. We're not there yet. We're in the middle of this research. So if you invite us back in 4 or 5 years, then maybe we can tell you if we were successful or not. But that's kind of one direction our research is going in the future. And I think this is back to you, Jenny.
Yeah, so we'll be wrapping up soon. So here, basically kind of summarizing some of what we've told you. So we really asked, how do plants adapt to different environments? They do so by cueing their germination to happen at the right time. And we found some of the genetic mechanisms that seem to be linked to these responses and how it varies across these species that inhabit different climates. We also know that they may adapt by simply staying dormant and not germinating, like our desert species, and our genetic results also suggest this is happening as well. We know that germination timing affects the cues they experience for flowering and their timing of flowering. Um, and so what is the impact of current and future climate change? Well, we're already seeing that some of these species are probably experiencing shifting life history timing, so shifting germination timing, which is then shifting when they're flowering. And we also see that for many of those species, that's reducing their seed output. Um, so then the next question is, can we develop strategies to mitigate climate impacts? Hopefully, as Julin said, we're working on that. Um, we're exploring the effects of doing that. And I think I also wanted to note here that the species we're studying are closely related to several rare and threatened and endangered species. So we're also really interested in, this is even further down the road, but understanding whether the knowledge we're gaining by these approaches and the jewel flowers we're studying is helpful for managing those other rare and threatened jewels in California. And next slide.
Sorry, yeah, trying to get a change.
And I'll just go through this very quickly. The other thing we're doing is we're using our barium specimens as a window into the past to try to understand what might happen in the future, because we've got these great collections that are essentially a window into what's happened in the past. We know where these specimens were collected and when, and we can also combine them with historical databases on climate, soils, and geography, and understand more about what shaped their responses in the past to try to understand what might happen in the future. And I think with that, we'll just acknowledge our collaborators and all the wonderful people that have helped us with these projects, and hopefully there's some time for questions.
Yeah, I also want to thank everybody for sticking with us for
Yes.
probably a pretty sciencey talk. So anyway, so thanks everybody for sticking with us. Yep.
All right. Thank you so much, Dr. Maloof and Dr. Gremer. We've got time for a few questions. And so first up, we've got one from Roxanne in the chat. She's asking, how important are pollinators to these species? If important, are there climate change factors that might need to be investigated relating to jewel flower success?
Chime in on this one, and Julin, feel free to add. Pollinators are really important for these species. We've studied this more intensively in the mountain jewel flowers, Streptanthus tortuosus. But generally across most, if not all of these species, I think all of them that we have worked with, there needs to be some pollinator or like at least some movement of floral parts around in order for seeds to be produced, for pollen to get on those on the stigma for reproduction. And so, yeah, I think we have to go in and hand pollinate these plants if there aren't pollinators around for them to set seed. So it is really important, and also trying to understand kind of when they're flowering and whether they're still synced up with flowering so that they can reproduce with each other, like mate with each other, is actually really important. They tend to be pollinated by generalist species, so that does help. Having generalists means that there might be a broader range of pollinators that can provide those services, but understanding how that shifting flowering timing is influencing whether those pollinators are finding them and whether they're actually getting those pollination services is like an open question that we're also very interested in.
Right, because like the timing, the response to changes in the climate can be impacting the pollinators and the plant species differently, right? Yeah.
Exactly. Yeah. A good
Right.
question, Roxanne. Well, we'll find out. Sorry.
Do you have any other questions for our speakers? You can either raise your hand and ask on camera, or feel free to put it in the chat. One question I had was how long are jewel flower seeds viable for in the soil? So if they're postponing germinating a year, how much of a window do they have? Are they viable for decades or just a few years?
Jenny, do you my impression is it's like probably just a few years, but Jenny may have more data than me on that.
We don't know. So I know this more from we've actually buried seed bags in the soil at their home environments for the Streptanthus tortuosus, the mountain jewel flower. And we know that they remain alive, remain viable for at least 3 or 4 years, but that's as far as we've looked. I really wonder, so some of the desert species like Colanthus inflatus, which maybe some of you have seen the desert candle when you've gone down to Carrizo Plain or other deserts during a super bloom or something. They have really thick seed coats. And so I don't know, but I wonder and think that maybe that helps them survive even longer in the soil. We might need another decade to tell you.
Jim's wondering if there's any influence of fire on germination.
I can say, I would say anecdotally, and then maybe Jenny, you can add more. I mean, so there has been fire that, so I know this most again for mountain jewel flower, and definitely some of the recent fires have gone through very near our field sites. And so you've seen actually kind of just anecdotally a lot of germination, a lot of plants, a lot of very happy plants in the years after those fires. So that's kind of the level at which I can answer that question, Jenny. I don't know if you have more that you can add to that.
That's what I can answer too. So yep, I would echo what Julin says. I will also say that a graduate student in my lab is really interested in understanding these cues and is doing a germination trial to see how these seeds respond to things like liquid smoke and this like nitrogen, which is often released as part of a fire. So also in like a few weeks or a couple months, we'll know a little bit more about that.
So to answer Wendy's question, Wendy says, we noticed that after some of the Coast Range fires, some Streptanthus grew to enormous size. What causes this removal of non-natives and fertilizing? And yeah, I think it is both of those. Less competition and like a really nice fertilizer in the soil. Exactly. Seraphima asks, are the pollinators for low growing populations the same as for the higher elevation plants? I don't know the answers. I can tell you that because we did some planting actually at UC Davis field sites, and we had a ton of kind of general honeybee pollinators there. But I haven't seen that many pollinators at the higher elevations. Jenny, you spent more time up there. I don't know what you've seen, but
I think the pollinators are slightly different. One of my grad students did more observations, so they still tend to be pollinated by kind of generalists like flies and bees. So there's some overlap for some species that are really pollinator species that are really broadly distributed. But yeah, they are pollinated, I think, by different species that can't can live at those different elevations, but I don't know more about how different those are.
I do think it's a really important question. I mean, there's like
Yeah.
so many things we want to do, and we only have so many people. But yeah, I mean, all these are really great questions.
Yeah, and if we introduce genes that are good for climate adaptation, does that change what their flowers look like or when they flower? And does that also influence their interactions with pollinators? Those are all really interesting questions that right now we don't know the answer to, but are really interested in finding out.
Mhm. All right, any other questions? All right, so I think we'll wrap things up there. Be sure to tune in for our talk in February, which is going to be from Dr. Glenn Holstein, who's going to be doing an overview of biodiversity in California. So that should be a good talk. So be sure to stick around for that one.